the elders past and present and emerging from that society. And also, I'm not sure what the traditional owners are in Albany, but also acknowledging them. I wanted to start the presentation by perhaps going over about how I came to be in the position I am at the moment. And so my career actually started with doing a course in pharmacy. So when I finished school, I was particularly interested in chemistry and biology, and I thought, pharmacy in particular was a wonderful way to sort of explore that. And at the same time, I had a cadetship with a chain of pharmacies called Sol Pattinson, which are fairly prominent in New South Wales. I don't know that they actually have any shops in WA, but I would work on my weekends and sometimes after hours, and I finished my course and I got my registration with Sol Pattinson, so I was a registered pharmacist, and then I guess promptly decided that retail pharmacy wasn't really the scientific endeavour that I was looking for, and so I decided that I would perhaps change tact a little bit, and I went up to Townsville in North Queensland and I started my undergraduate degree all over again, and I focused on marine sciences and zoology, and I did my undergraduate again, and then I did honours, and I did it with this fellow who's crazy hair, a little bit like my own crazy hair. And he was interested in looking at herbivory. His name was, his name still is, I believe, Professor David Bellwood. And he is interested in herbivory and the functional morphology of fish, how fish actually accrue or collect that herbivorous material or the algal material from the reef, how they digest it, how they assimilate it, what are the ecological impacts of that. And so I did a project with him looking at these fish here, the damselfish at Lizard Island, which is in the top pane there. And then that evolved into a PhD project looking at diet and herbivory, and in particular the importance of detritus in marine food webs, but this time looking at blennies, which are these little fish that you see on the bottom part of the slide. And then after I'd finished my PhD, I was fortunate enough that I was a registered pharmacist, so I still was working on weekends and times, so I had plenty of money. But what I'd missed out on was the teaching of students and the demonstrating, and so I did a little bit of that once I finished my PhD, and then I was fortunate enough to get a job in the Caribbean, and this involved basically mainly American, well, pretty much exclusively American students coming down to this island in the Turks and Caicos Islands, which is just to the north of the Dominican Republic. And we'd spend a semester. In the mornings, we'd talk about ecology and fisheries and socioeconomics, doing lectures there. And then in the afternoon, we would actually go take the students out into the field and do snorkeling exercises or sometimes scuba diving exercises, where we'd initially do IDs of fishes and corals and algae. But then the students also had an opportunity to develop their own little projects that they would work on for the rest of the semester. And that was great for a couple of years. So I lived actually on site, this fairly remote location in Caicos Island. And I then got an opportunity to work in the UK as a postdoctoral researcher at Newcastle upon Tyne, which is in the northeast of England. And there I met the guy I was working for on the top right is Professor Nick Palunin, who at the time was the president of the International Coral Reef Society, so quite an influential fellow with lots of connections all around the world, which was fantastic for getting money, but also getting to different locations around the world. And he had a student, this guy in the bottom left, Nick Graham, who was doing his PhD and who I started to work with in 2005 and continue to work with today as like one probably my greatest collaborator. We did some studies in the Seychelles and continue to do work there, looking at the effects of bleaching on fish, but also how that affects the fisheries, things like nutrient transfer, the vulnerability of fish in terms of whether they're rare species or and whether they're more susceptible to things like bleaching or fishing, and the effectiveness of marine reserves in protecting fish and corals, which is an interesting question in its own right. After that, I moved back to Australia after that postdoc was finished, and I worked for DBCA, who I'm sure you're all aware of because they're fairly prominent down in the south in Albany. And they're a little bit unusual, DBC, and as a government organisation, in that they have an actual science group embedded within the department. And so I worked for the Marine Science Program, whose remit was to do monitoring and research for the network of marine reserves that occurred around the state, and then also to look at the threatened and protected species, so things like the turtles and dugong. And so we had a big program. My role was really in terms of doing research rather than monitoring. So I'd look at the appropriate ways of actually doing the monitoring, so helping with different methodologies and working out whether they're compatible, new emerging technologies with the existing ways, taking that data and looking at some of the long-term trends and things like the effectiveness of sanctuary zones. So that was really interesting time, but I found as I was getting towards the end of my time there, I was being more and more pushed into the management side of things and less about the research, which I wasn't that happy about. And so a couple of years ago now, I took up a job at the Australian Institute of Marine Science. And the AIMS, or Australian Institute of Marine Science, is much more at a Commonwealth level. So we have about, I think, 500 employees. Most of those are over in Townsville in North Queensland, associated with research on the Great Barrier Reef. But we also have about 100 or more people in the Perth office that work on a number of different things, but predominantly on the northwest part of Australia, and mainly in the tropical sense of things. So have amazing infrastructure, things like the boat, Solander, an amazing group of scientists here as well. Been working on things like understanding seaweeds and how effectively they capture carbon, and then whether that carbon is actually sequestered into sediments. Looking at nutrient transfer between different systems, so how important things like nutrients that come from seabirds are in terms of depositing that on islands, and then are those nutrients then important for flowing back into the marine system and increasing productivity there. And I've continued my work to date on things like coral bleaching and the impact that that has more broadly in terms of fish and fisheries and just general ecology of the marine system. And that obviously is what I'm going to talk to you a little bit in more detail today. So you've probably seen figures like this before. This is something that I took straight off the Bureau's or BOM site, and it's from last year's State of the Climate report. And it shows in both the land and the sea, we've had this gradual increase in temperature. And from the start of the last century, or over the past more than 100 years or so, there's been an increase of about 1 degree in terms of sea surface temperature. And that might not seem like a huge amount when you think about it, but you need to remember that that's averaged across all of the Australian waters. So, and it doesn't take into consideration that now when we have these extreme events like marine heat waves, we're already sitting 1 degree above where we normally would have, or where we would have been 100 years ago. And it's especially sort of relevant at the moment. You may be aware that this summer we had a really extreme or intense marine heat wave across the northwest of Australia and even flowing down into the south as well. And this is on the top left here a map of Australia, and it's just showing you how far above the normal sea surface temperatures we were in places like Ningaloo and the Pilbara. Sometimes it's like 3 or 4 degrees above the average. And people have actually sort of roughly worked out what that sort of stress means for something like a coral. So corals are living very close to their ecological threshold in terms of where too much heat can cause stress. And so what they do is they calculate what they call a degree heat week. And a degree heat week is essentially, if you have the summer maximum temperature, every time the temperature goes above 1 degree of that, the maximum sort of temperature that's been averaged over a long period of time, then that's 1 degree heat week. If it goes 2 degrees above that, then it's 2 degree. Or if it stays at 1 degree for 2 weeks, then that's 2, or 3, that's 3 weeks. And generally what we see is if we get to 4 degree heat weeks, then we start to see the corals get stressed and they start to bleach. And if we get to 8 degree heat weeks, then those corals actually start to die. And unfortunately, this summer is probably, it's the most intense sort of heat stress that we've seen along the coast of northwest Australia. So places like the Pilbara and even Ningaloo have experienced up to 20 degree heat weeks. And these pictures that I have here on the right are just some of a couple from Ningaloo, and you can see that the corals are quite pale. And in some cases, the brown that's around the branches is where those corals have actually already died. Like quite sad and intense event that's unfolded this very summer, and we won't really know the full extent of that event for probably another sort of 6 to 12 months. So I've already mentioned coral bleaching, but I should probably take a step back and give you a better idea of what coral bleaching is. And when I was putting this together, I was reminded of a talk that I went to a long time ago where there was a Japanese scientist and he described corals as a monster, which I thought was pretty cool. He said that the monster was a rock, and then over the rock there was this thin veneer of living tissue with hundreds and thousands of mouths, and those mouths were pulling plankton and particulate material out and sieving or filtering it out of the water column, and that was their source of food. But at the same time, these filter feeding monsters also had within their tissues little microscopic algae that could photosynthesize and produce their own energy, which could then be used by the corals. So you had this combination of heterotrophic feeding, which is the filter feeding component, and autotrophic feeding, where you have these symbiotic algae or zooxanthellae that are living within the tissue of the coral. Unfortunately, what happens is when the coral gets stressed, and it can be from all sorts of things, it might be through changes in salinity or too cold conditions, but the most common form of stress that we hear about with corals is heat stress. And so when they get stressed and they get to that 4 degree heat week level, the relationship between the zooxanthellae, or the little microscopic algae, and the coral starts to break down. And the corals expel that algae, and because the algae gives the corals its actual colour, then the coral starts to look white, and that's why we often refer to that as bleaching. Now, because the actual tissue surrounding the skeleton of the coral is still alive, then the corals can survive even after bleaching. It doesn't necessarily mean they're going to die. Unfortunately, if that stress goes on too long, then they will die. the tissue around the skeleton tends to slough off, and the skeleton is then occupied by things like turf algae, which is what's shown here on the right. So different types of corals seem to be more susceptible to this heat stress and bleaching than others. So some genera, which I assume you're familiar with the idea of genera, so things like the acroporids and the posloporids seem to be very sensitive to heat stress and bleaching. Different growth forms, so things like plating corals up here on the top right, and corymbose or branching corals also very susceptible to bleaching, whereas things, and it's not a great photo, but massive corals, this one in the very bottom left corner, they tend to be a little bit more resistant or resilient to heat stress. It can also, whether corals bleach or not and they survive, can have to do with the types of zooxanthellae. So some species of zooxanthellae just seem to be more resistant than others in terms of heat stress. Similarly, the type of coral can be genetically adapted to heat and better able to survive heat stress than others. And of course, as I mentioned before, there's two modes of feeding for corals. So some species of corals are a lot more dependent on that heterotrophic or filter feeding pathway than the autotrophic component. And those species or those types of corals tend to be a little bit more resistant and able to, survive heat stress and bleaching than those that are heavily dependent on the microalgae in their tissues and are more autotrophic. So what does this all mean, though, for the fish or for the broader ecosystem when we have a bleaching event? So up here, I've sort of done a broad sort of graphic of a coral reef, and in yellow is what you'd expect from a healthy reef. And then in the background in black is what might be left after an extensive bleaching event. And then to the right of that figure, you can see in yellow again, the corals may recover. And in some cases, it'll depend on the severity of the bleaching and the type of corals. That can take 7 to 10 years for a full recovery, sometimes longer if it's in a remote location and if the bleaching has been really extensive. And then in the right, I've just put a few bars of very broad trophic or feeding groups, and the pattern that you would expect to see for each of those, and the width of those bars also, as a general sort of estimate, gives an idea of how abundant they are within the fish community. So you can imagine that when you have loss of coral through bleaching, the types of fish that are going to be impacted the most are the fish that feed on corals, so the corallivores. And there's lots of different fish that do that. Well, there's not lots, I should say, but there's a few different species. The one that most people are familiar with are different types of butterfly fish, and that's what we've got here. Common name of this guy is the melon butterfly fish, but feeds on coral, and numbers of these fish typically decline when we have a bleaching event. On the other hand, we've got the herbivorous fish. So remember I said when the coral dies, the skeleton tends to be occupied by this filamentous or turfing algae, which provides food for these guys. So things like this, this is common name as a convict tang, which is a type of surgeon fish, very common at places like Ningaloo, occur in big schools, go around and feed on that turfing algae that occurs on the dead coral skeletons. Their numbers tend to increase after a bleaching event, and then as recovery of coral occurs back, then their numbers decline back. So this guy here, so this is a monocle brim, and it's representing invertebrate type species. So their numbers seem to stay fairly stable, and that's because their food source, the invertebrates, if anything, stays the same. There's just as many invertebrates, so little crabs and worms and the like, within the rubble and the dead coral as there is within the live coral and the turf algae. So they seem to be quite fine. The other group that sometimes doesn't do very well is the planktivores. So in particular, the very small planktivores, which are often up in the water column, picking plankton out of the water, things like little copepods and the like. And the reason that they don't do so well is that a lot of them actually tend to take shelter in live coral, which is exactly what these little chromis or little damselfish are doing now. So when the corals bleach, they essentially lose their home or their form of shelter. And there are larger planktivores which seem to be a little bit more resilient, so they're able to survive these bleaching events, but it's the small ones that are coral dependent that really seem to suffer. And then finally, the piscivores, so the fish that feed on other fish. And this is the example I've got here is a peacock grouper. But what happens with those guys is initially we see a little bit of an uptick sometimes in their abundance because their prey becomes easier to catch, things like those little planktivorous feeding damselfish are much easier for them to feed upon, so they have an increase in prey availability once they don't have their shelter, and then the numbers tend to go back down after that sort of initial availability of prey increases. So they're general theories about what we might sort of expect in terms of the trophic structure of the fish assemblage and what happens with a decline in coral due to a bleaching event. I must say, like, I'm talking here strictly from bleaching event, there's also potentially direct effects of the temperature on the physiology of these fish as well. So we're talking about the indirect effects of habitat loss here, is often how it's referred to in ecology. If we now look at actual species, and what I've got here is a bit of a figure which is from what we often refer to as a meta-analysis. So we've taken lots of different studies and we've averaged the response of these different species from all of those studies. And on the y-axis, if we have negative values, that means with a loss of coral, then we get a loss of that particular species. If we get a positive value, which we've got on the right, then those species tend to increase in abundance. And I've color-coded that here, so the corallivores are in red, so those fish that are coral feeders, and the coral dwellers, so a lot of those are, the planktivores are yellow, and the herbivores are in green. And typically the patterns, as I mentioned, for the broad trophic group, so herbivores are increasing most of the time, not always. Almost always the corallivores are declining, as are the coral dwellers. The interesting thing here, and I sort of bring to your attention, is that the extent to which a species declines is highly variable. So in some cases, there's hardly any decline in the corallivores, so this species here in the middle of the plot, and in some places, or for some of the species, they basically go down to nothing, totally decimated. So the question is, why might that happen? And the reason is to do with what we sometimes refer to as ecological versatility. And at its broadest level, we can break that down into obligate and facultative dependence. So if we go back to talking about butterfly fish, here I've got 4 different species of butterfly fish. On the left, we've got the chevron and the melon butterfly fish, which I mentioned before. They're obligate corallivores. All they ever eat is coral. So they're obligates. On the right, we have a raccoon and also a threadfin butterfly fish. They're often referred to as facultative corallivores. They will eat coral when it's there, but if the coral's not there, they seem to be able to survive. So they can feed on other invertebrates or even algae in some cases. And as you'd expect, those that are obligate corallivores with a decline in coral, their numbers decline. Those that are facultative, that are a little bit, able to change their diet as resources change, they decline. The extent of their decline after coral loss is nowhere near as severe. And in some cases, their numbers can either stay the same or rarely they actually increase because they then start targeting the invertebrates. We can take it one step further than that as well, because even within the obligate feeding butterfly fish, we have specialists. So remember this one here, the chevron butterfly fish, feeded on Trophacealis. That species tends to only feed on one or two types of coral, typically big plates of Acropora, which unfortunately for Trophacealis is, as you remember me saying earlier, one of the types of corals that's really susceptible to heat stress and bleaching and dying. So because it's such a specialist, what tends to happen is its numbers, which this will be Trophacealis over here, its change in abundance is very negative. Whereas with Lunulatus, or the melon butterfly fish, it will feed on maybe 30 or 40 different types of coral. So even though its preferred food, which might be the plating coral, is gone, it's able to switch its diet and then start feeding on something else. And so typically the extent of change in their abundance is nowhere near as high as it is with Trophacealis or the chevron or the highly specialized feeding fish. And we see this same pattern in other types of coral dependent fish. So these are all independent studies. One is for gobies that live in corymbote, little sort of acroprid corymbose corals. Those that only use one of those corals, or the little damselfish, those that only inhabit one type of coral, they always are more susceptible and have a greater extent of population decline than those that are able to use multiple species or types of coral. So that's this idea of ecological versatility, and that specialists tend to be a lot more prone or susceptible to disturbance than those that are generalists. If we go back to this figure that I showed before with the meta-analysis, you'll note that there's some species that seem to decline even though they're not dependent on coral. And I've just circled a couple there. And there's a couple of reasons that this might occur as well. So one might be that we often think about fish only in their adult form. But of course, when they first come to the reef, or to a coral reef system, they're very small, and a number of these species will hide and live in the coral as juveniles, even though as adults they don't seem to have any type of affinity or association with coral. And a good example of that is this guy down here with the yellow head and the white body. So as a juvenile, it tends to be very closely associated with finely branched corals where it takes shelter. But as an adult, it's a fairly drab black sort of fish, a little damselfish. And although it feeds occasionally on soft corals, isn't always closely associated with coral. So part of the reason that species may have declined is because the juvenile habitat has been lost. And there's potentially a bit of a lag effect before we see an impact on the adult population. Another reason, and probably from my perspective, a really important reason, is that the corals themselves are providing structure. And so if we have a coral reef like on the left there, and then eventually over time after the coral dies, that coral breaks down, either due to physical action like the waves or tide action, or we have biological action, so feeding by parrotfish and urchins breaking down that structure, we lose a lot of the structural integrity of the reef and a lot of the refuge holes and spaces for fish to actually hide in. And that can have a much broader impact on the overall fish community, because that starts to affect all of the fish that that require some sort of shelter on the benthos or within the reef system. And so what we tend to see on the right here, when we look at our bars of abundance for the different trophic groups, is now we get a decline in coral feeding fish. They don't come back. Also the invertebrates and all of a broader array of the planktivores and the piscivores. The one thing that tends to still do all right to some extent is the herbivores, although they will also decline due to a loss of structural complexity as well. They may continue to increase if we get what we call a regime shift. So if that coral is now replaced by fleshy brown, usually macroalgae or seaweed, then that structure is replaced, that was once coral, is replaced by the actual algae. But it's a different type of structure and a different type of environment, and that comes with a totally new type of fish community. And so the herbivorous component there are often what we refer to as browsers. So they're very specialized herbivorous fish. So some of the rabbit fish, some of the kyphosids or chubs, and some of the unicorn fish are very specialized on feeding in that, and they can benefit from that type of environment. But obviously we've lost all of the corallivores and some of the invertebrates and planktivores and even piscivores. And we can actually, because structural complexity is such an integral part of the reef and drives a lot of the processes that, or is the underlying sort of measure that drives process on the reef. It's one of the things that we're really interested in measuring, and there's lots of different ways we can do it. In more recent times, people are using photogrammetry, so we would take loads of photos of the reef and then essentially build a three-dimensional model, and then we can measure the amount of holes and refuge and space there for fish and other things to hide. In the old days, what we used to do is get a measure of rugosity, and in the top left is how we might do that. Often we'd get a length of chain, say 5 to 10 meters long, and we'd carefully drape that over the reef and make sure it's tucked into all the little nooks and crannies and holes and the like. And then we'd get a tape and measure how far linearly that distance has gone. And you can imagine if it's a very complex, highly structured system, then it's not going to go very far. So the ratio of linear distance to fixed or conformed distance gives us a measure of rugosity. And that measure of complexity, whether it's through rugosity or through photogrammetry or even coarse measures like the height of the reef and how much that changes, that tends to be directly related to the biodiversity of the fish assemblage. And biodiversity in its coarsest way can be measured as species richness, but there's also diversity measures like Simpson's and Shannon-Weiner metrics as well. They tend to all behave the same. So as structural complexity increases, the diversity of the fish assemblage also increases. So if we have an event like the loss of all of these branching corals through a bleaching, for say, and we get these rubble fields where a lot of the complexity is lost, and we've seen this in a number of places around the world, the diversity of the fish assemblage is also compromised or lost. So what happens in terms of overall change? So again, I've got our little sort of map or diagram of the reef going from a healthy system to one where the coral has died but the structure is in place. Then the structure breaks down, and then sometimes that structure might be replaced by macroalgae, so sort of big leathery things in the tropical environment. Those things are like sargassums or turbinarias. So diversity initially high. Diversity can be retained and still remain high even after the coral has died, if the structural complexity is also retained. And that's because a lot of the fish that are dependent on coral are replaced by other species that are benefited by having greater abundance of turf algae. If that breaks down, that structural complexity, and you end up with rubble beds, then you also have a loss of the diversity of the fish assemblage. Then it might increase as the overall complexity increases again with a change in the foundation species. So some of the macroalgae, so sargassum, or although it doesn't really occur on coral reefs, things like kelp. And I've already sort of talked about, what this means for the broad sort of trophic groups, a shift from a coral and planktivore dependence sort of coral fish community when the corals are healthy, to one that is increasingly dominated by generalists and also species that benefit from higher abundance of algae, so the herbivores and some of the invertebrates. What this means in terms of the food web is we tend to move from a system which is based on pelagic sources, so corals, like I mentioned earlier, are partially filter feeders, but you may have heard corals being things like the wall of mouths. So at the front of the reef, lots of corals, lots of filter feeding components, lots of planktivorous fish that are continually picking energy in the form of plankton or particulate matter out of the water column and pulling that back down into the reef system, where that now energy drives trophic pyramids and trophodynamics. But if we move to a system where coral is no longer available and increasingly macroalgae and a loss of planktivorous species, then the system shifts to one where the food web is driven by benthic rather than pelagic resources. And what does that mean for the overall fishery? So in a reef system, the fishery might be dominated by things like some of the snapper and some of the labrid. So not so much in Australia, but particularly some of the scarids or parrotfish, and also some of the surgeonfish. If the structure is retained, then the fishery is usually still fairly productive. In fact, the loss of a little bit of structure can actually make the fishery even more productive, because the predatory fish can actually then access their prey, and it tends to, at least in theory, increase the productivity of the fishery. If you get a system which is completely flattened and you lose that structural complexity, or a system like that, then the fishery tends to suffer the productivity. You lose a lot of the fish, and that can be really detrimental. But if you move to an algal system, then the fishery may still be okay. It's just that the type of fish that are targeted become completely different. So in places like Ningaloo in Australia, the things like the emperors, so spangled emperor and some of the cod, are really pretty much heavily dependent on this macroalgal environment. And so they can become an increasingly important part of the fishery. In other parts of the world, things like rabbitfish tend to recruit and live their lives in the algal environment. So really important for the fishery there. And ideally what you have is both systems, because that gives you some insurance. If you have healthy coral reefs and healthy algal systems, then you have a fishery that isn't dependent on just one particular system. So ideally you have both. But we have seen in some parts of the world where you can have extensive coral loss and the fishery can still remain productive, especially when it's dependent on species that are algal associated. It's a lot riskier over time because now you've lost some of your redundancy in the fishery system. And with that, I think I'm pretty much done, and so I will open the floor for questions.